Hi everyone, this is the second lecture in Chapter 6, which is all about microbial nutrition. Today I'm going to talk about environmental factors that can play a role in how microbes get their nutrition. So when we look at environmental factors and we're talking about microbes, hopefully you can imagine that there's a great influence from that, all the environmental factors that microbe lives in. Okay, and these environmental factors can affect the function of enzymes in the microbe. They can affect what nutrients are available for the microbe. And so survival in this changing environment that they live in, since they don't really control their environment, is largely a matter of whether or not that microorganism has an enzyme system that can adapt to changes in its environment. They don't really have a control over that environment, so hopefully they have the ability within the cell to break down or utilize things that might be found in the environment. So the first of the environmental factors that we're going to look at is temperature. So we have what is called the cardinal temperature, and that's the range of temperatures that a given microbial species can grow in. There's obviously a minimum temperature. It's the lowest temperature that that microbe can still have growth. And below that temperature, its activities are either limited or it may actually not survive. And then we have a maximum temperature, which is obviously the opposite of that, the highest temperature that the microbe can still grow and metabolize without its proteins becoming denatured. And then each microbe for its individual species will have an optimum temperature. And this is somewhere in between that minimum and maximum temperature where you have the fastest growth rate and metabolism rates. So when we look at this figure, this is in your book, and this is a really good one showing the different temperatures that you might see some of these microbes at. This is figure 6.5, and we see a bunch of different groupings of microbes. So the first one on the left is the light blue line, and these are called psychrophiles. These are ones that its optimum temperature is really cold. It's usually right around in between 5 and 10 degrees Celsius, and it can actually thrive at 0 degrees, which would be freezing, the point at which water freezes. And these obviously you would probably find in like freshwater lakes, things like that, in temperate climates where it gets very cold. So those are the psychrophiles, and actually that cold temperature is actually what would incubate them. So if you put them into a freezer or something, they would actually be incubated there. When we move into the yellow kind of mustard colored line, this is called psychrotrophs. These grow slowly in cold, and their optimum temperature is really cool, kind of about the temperature of a refrigerator. So some foodborne illnesses that we have in humans can be found from a group of bacteria that are actual psychrotrophs. And one of those is listeria, and it actually, in the refrigerator, can still reproduce and multiply. Now the green line is what we call mesophils, the kind of the middle temperature ones. And their optimum temperature is right between really 30 and 40 degrees Celsius. These ones tend to be the most medically significant with humans, and that's because our body temperature is 37 degrees Celsius. So they actually really thrive and grow quickly at a temperature that our body is at. So these are the ones that we normally would think of as human pathogens that do really well in the human body. The pink line then is what we call thermophiles, loving kind of that higher temperatures. And the thermophiles here, you can see the optimum temperature is right around 70 degrees Celsius. These are very, very warm. And then what we have lastly is this maroon line called extreme thermophiles. And these actually are at extremely hot temperatures. You might find them in volcanic areas, maybe in very, very hot springs water. So these are called extreme thermophiles. But as you can see, there's a range that each of these lives in. And so if they get outside of that range, it actually ends up being a way to control them. So hopefully that makes sense, and why we use really high temperatures for medically significant pathogens. Generally speaking, that should kill them because we're past their maximum temperature. So after temperature, one of the other environmental factors that we often talk about are gases. And the main one that we talk about is actually oxygen. There are a few that really like CO2, but when we talk about gases as an environmental factor, and what you'll see in lab oxygen is what we look at. So oxygen has the greatest impact on microbial growth, and the microbes will fall into three categories. There are those that can use oxygen and detoxify it. And then there are those that can not use it nor detoxify it, so it's actually will kill the microbes if they're exposed to oxygen. And then we have those that don't really use oxygen. It's not what they use to grow, but they can detoxify it, so they can still grow with oxygen present, they just don't utilize it. Okay? When we look at these, this is something that you should be doing in lab or should have just done in lab. And this is a table on page 153. It's table 6.5, and this is a great one showing the different oxygen usage patterns in microbes. Hopefully you've seen these in lab by now. The first one are what we call the aerobes, and they use oxygen. They actually need oxygen, so they're obligate aerobes, meaning that they can only grow in the presence of oxygen. So if we have this test tube with the solid media, all of your growth will be right at the top where the oxygen is found. Okay? The next one that we see here is called microaerophils, and they use oxygen but at a lower atmospheric concentration of what you would find in air. So what you see is you'll get this little kind of area above of medium that doesn't have growth, and then a band of growth where there is still oxygen getting in but not a lot, and then no growth below it. Okay, so those are microaerophils. The next group is called facultative anaerobes. So they don't require oxygen for metabolism, but when it's present they will use it. So in this one what you'll see is that you'll have growth throughout, but the majority of the growth will be in that upper part, probably upper 2/3, and that's because growth in the presence of oxygen is actually faster than anaerobic growth. So that's that facultative anaerobe. Our next one is the anaerobes, and these are obligate anaerobes, meaning that they cannot grow in the presence of oxygen. So they need an anaerobic environment to grow. And what you'll see here is you'll have a large portion of the top part of the medium without any growth. And then about halfway down where there's no oxygen present, down to the bottom, you will see all of the growth. And then the last one that we have is called aerotolerant anaerobes. These do not utilize oxygen, but they're going to be able to survive and grow a little bit within its presence. So they're not really harmed by oxygen, so you'll see growth throughout, but it will also be a large amount of growth at the bottom. It'll be the same from bottom to top, whereas with a facultative anaerobe, you would see less growth at the bottom. So hopefully this makes sense on these oxygen usage patterns, and you've seen this in lab. If you have more questions though, don't hesitate to ask me. So that was on the gases influence, environmental influence on microbes. The next one that I want to talk about is pH. So when we talk about pH, it's on a range from 0 to 14, 0 being the most acidic and 14 being the most basic. So kind of that neutral area is the pH of pure water, which is 7.0. And the majority of organisms will live or grow where the pH is between 6 and 8. And that's because some strong acids and bases can be damaging to the cell. There are some that are acid loving or alkaline or base loving. They're kind of more rare, so the majority that you would see would grow in that 6 to 8 pH. We also have an environmental factor of osmotic pressure. So osmophils live in habitats with high solute concentration. So going back to that lecture that I did earlier in this chapter, this means that there are lots of solutes in the liquid surrounding the bacteria, which means that the water from the bacteria will want to move outwards. It wants to leave and kind of shrink that cell. So we have organisms that are called halophiles, which love high concentrations of salt. We have some that are obligate halophiles, meaning that they need to have at least 9% sodium chloride. So they need salt to actually grow, and they can grow really well at 25% sodium chloride. So huge concentration of solutes outside the cell actually is needed for these types of microorganisms. We also have facultative halophiles. They are fairly resistant to salt, even though that's not their normal environment. So they can actually be okay in that osmotic pressure. And one of those that you might see in lab is Staphylococcus aureus, which can grow in sodium chloride anywhere from 0.1% up to 20%. So they actually have a very good means to survive that high salt concentration. Another environmental factor that can affect microbes is radiation. Now for phototrophs, this is actually where they get energy source. They're going to get it from the light rays. And then we have non-photosynthetic microbes that can actually be damaged by it. So there's both negatives and positives. The phototrophs actually love radiation, and many of the other microbes, this is actually can be damaging to them. Some microbial species will produce carotenoid pigments to protect them from it, but ultraviolet and ionizing radiation is something that is often used for microbial control to kill the microbes, since most of them are not phototrophs. And then we also have the environmental factor of pressure. So when we think about just in general microbes, we're generally thinking about the atmosphere we live in. But there are microbes called barophiles, which exist under pressures that range from a few times to over 1,000 times the pressure of atmosphere, so what we live in a daily basis. And these would include deep sea microbes, which are really adapted to that high pressure of the ocean, and that if they are brought up and exposed to normal atmospheric pressure, it will actually burst them. And then the last environmental factor I want to talk about today is other organisms. So if you can imagine bacteria in a natural setting, they are living in shared habitats. And this is with other microbes that might be similar or dissimilar. They can also be associated with different kinds of multicellular organisms, animals and plants. You think about the microbes that live within our body or those that live out in nature with plants growing. And their interactions can be one of a number of things. They can be beneficial. For example, we have bacteria in our stomachs that are beneficial and help break down things that we can't. There can be bacteria that are harmful. You can see that in both plants and animals. And then there are bacteria that basically have no particular effect. They're ubiquitous, they're found everywhere, but we don't really notice them, they don't harm us, and we don't do anything to them. So these interactions can be either obligatory, where they need the interactions to survive, or non-obligatory, where they're fine with or without them. And most of the interactions with their environment and other organisms is going to involve nutritional interactions. Perhaps they're getting nutrition from the organisms they're associated with, or perhaps they're competing with those organisms. So you can imagine that can affect their growth. So hopefully this gave you a little bit of insight into environmental factors that affect organisms, microbes here. Hope that was helpful. Thanks.